Good morning, everybody, and welcome again to another episode. Please bear with me. I don't have fancy sound or equipment or things like that. So this is just a very basic, down-to-earth sharing of information and helping the grade 12s in South Africa get their heads around the curriculum material for life sciences and biology. Hope today is going to be helpful to you guys. I'm going to be doing genetics. We're going to cover as much as we can in one video, but I'm probably going to divide it into more than one session because there's a lot of work to get through. So we're going to start off with a basic introduction to genetics. And also, I just want to let you guys know I've created a separate video on the SAM School Biology channel for all the terminology and definitions thereof. It literally runs through all of the key terminologies and definitions found. I'm working from the Nelson Mandela University textbook that they compiled to support the grade 12s in this subject. And that's really available online. You can download it and access it for yourselves. So they've created a very nice, simple, easy to understand, comprehensive overview of the entire syllabus for the CAPS curriculum, but it's good for anybody really. So to get started, guys, I'm not going to go over the key terminologies again because, as I've said, that's available in a different video. But let's start off with our introduction to genetics with Mendel as the father of genetics. Now, also, as I go along in the video, I'm going to comment as I feel that there's a relevant exam question, because remember, a lot of the time we're going to have a perfect exam type question and we might not always recognize that. So I'm going to help you guys think about when you're reading through your stuff and studying, think about this as a potential exam question. In other words, think about it as if you're the examiner. Would I ask this as a question? And also, I always say this in the videos that I'm doing, please, guys, have diagrams and sketches and pictures to use as reference, because when we're studying biology, we always want to have something to look at to complement the work and the information that we're looking at. Okay, we want to have something to look at because it just helps. It makes biology that much easier. Okay, so let's get going. I'm going to share a page with you guys from the. Oh dear, sorry guys, that's actually the wrong page. Got to bear with me. I'm still new to all this live streaming stuff. It's quite a learning curve, so be nice, be kind, bear with me. Okay, there we go. Should be that now. We start off with our introduction. We're going to be talking about how genetics follows on from the sections you should have done previously, which was DNA, your protein synthesis, meiosis, and sexual reproduction. And once you've understood those segments, it leads through to genetics, okay? So please make sure, if you're not up to speed on the DNA, protein synthesis, and meiosis, sexual reproduction, go and review those first before you try and tackle the genetics. I haven't done a video yet on meiosis, but I'm going to be getting to those as soon as I can. So you can come back and check in and I'll have to cover those as well. So to start off with, genetics is the study of hereditary, okay, or heredity. So this means simply that there are going to be parents, whether it's animals, insects, organisms as parents, and they pass information on to their offspring or their children. And this continues. It's a repetitive cycle. So we know that every individual, let's talk about humans to keep it simple, every human inherits a set of genes, okay, which are found in your chromosomes. You get some from your father and some from your mother, half to be exact. And that's what makes us unique, because there's a combination of these things that come about to create a unique individual. We are similar and we can be identified as part of or related to, but we are unique. Okay. We've got the key terminology there. I'm not going to go through that because there is another video covering that. So let's move on. Those are the terminologies you can find in my other video. Also, the Nelson Mandela gives a lot of activities and answers in their curriculum. So you do well to go and download that and have a look at it, because it's really useful, and I don't cover activities in these videos at this stage. Okay, so Mendel, we know, is the father of genetics. He was Gregor Mendel, an Austrian monk And he was regarded as the father of genetics because of his work on garden pea plants, which helped to explain how genes are passed from parent to offspring. So remember, in his day, when he was doing his thing, they had no clue. They really didn't understand all of this. So his work was very pioneering. And he started basically observing these plants to see what traits were inherited. Now, remember, a trait is a characteristic or an appearance of something. So we're going to also discover as we go along that we get genotypes and phenotypes. So when we're talking about traits, we know that it's our genetics that bring those traits out in us, but what we see is the phenotype. So genetics or genes would be the genotype, which is the internal cellular structural stuff, and the phenotype is the physical or appearance side. So why do we have brown hair? Why do we have black curly hair? Why do we have blue eyes? Why do we have green eyes? These things are passed on, and these are the traits that we inherit. It's not just random. It's going to have to come from a mom and a dad very specifically. Okay, so we see that the traits with Mendel were, when he was talking about the peas in particular, he was looking at the shape of the seeds. He was looking at the color of the seeds, and then he was looking at the pod that contained the seeds. He was looking at the pod color, and then the flower color, also the flower position and the stem length. These were the different traits or characteristics that he was studying when he started his experiments and trying to figure things out and understand things. Okay, so a nice exam question that I always like to ask, it's normally a multiple choice for 1 mark, and they say, right, Gregor Mendel, and who was he? Okay, so here's a very nice table summarizing, and again in picture format, because remember I always talk about let's do pictures, because pictures are really helpful. So here we see that there were 4 different things, and then there were different characteristics or components. For the seeds, we had the shape, where we had a round seed or a wrinkled. For the color, we either had a yellow or a green seed. And then for the pod, again, we had the shape, which was going to be a full or a constricted, shriveled up, and then the color was either green or yellow. In the flowers, he had the position, which was either axial or terminal, and of course, purple or white. And then it was either a short plant or a tall plant. So these were the things that he decided to study, and he then went on and did a very controlled way of studying this. So in his experiments, Mendel noticed that garden peas would occur in 2 different heights, simply tall or short, and he chose to represent it with capital T and a short t. And peas are self-pollinating, so tall peas tend to produce tall peas, and short peas produce short peas. Mendel's first genetic cross involved tall peas cross-pollinated with short peas. So he kind of went and fiddled a little bit and, decided not to leave things to their natural course. And he went and did a controlled setup where he then cross-pollinated what wouldn't normally have happened in nature necessarily. The result was that in the first group of offspring, remember we have the parents that are going to combine to do the fertilization and produce babies, or the first offspring, the F1 generation. In that first situation that he experimented with, the result was only tall peas, which would have been quite interesting at the time, not knowing what we now know. So Mendel took the F1 peas, and so he had now taken the original parents away. He took the resulting babies, or the children, if you like, the F1 peas, he crossed them with themselves. So he interbred those generations. And he then produced what was called the F2 generation, which is the second group of offspring. The result was quite surprising, I think. Tall and short peas, but in a very specific ratio of 3:1, which means for every 4 plants, he ended up with 3 tall ones and then 1 short or dwarf plant. So this experiment illustrated as below, that we start off, if you see in the top block here, we've got the parent generation. That was where he crossed the original tall plant with the short plant. On the right, you can see a bigger plant with 2 capital T's representing its genes. So remember how it looks physically as its phenotype, but its genotype is represented with capital T, capital T. Those are the genes that are going to decide if the plant is tall or short. And then he had so these were the purebred plants, if you like. There was no crossing here at this point. He took his small plant, which is small t, he crossed those 2 together, and he got that F1 generation, that first resultant offspring. Now, what came about was that he found in that first generation, all of them were actually tall. Notice there in the block it says genotype, that they are all capital T, small t, and we will get into this detail more later if you're still confused how this works. But the genotype, or the genetic level of these plants, was a tall t and a small t, or a capital T, sorry, and the phenotype, which is how they looked, all the plants came out tall. But what was interesting is they were all carrying the potential now because they'd crossed, okay? So now you can see here, this is going to be the new parents, okay? Because we're going to take this generation and then cross-pollinate them. So he removed that first generation. He didn't now bring in the purebreds, if you like. He took these cross generational parents and now he crossed them again. So then he created the F2 generation. And again, if you have a look at the genotype, that's what's happening at a cellular genetic level. The phenotype is what came out of the ground and how the plants looked. Okay, so we had tall plants, but surprise, there was one little plant sitting there. So there were 3 tall plants for one small one. And this was his initial experimentation, which was quite, I'm sure, funky at the time. Okay, so he developed a law of inheritance. Let's go through that quickly. So Mendel formulated his first law of inheritance. So obviously these are awesome exam questions, guys. Make sure you go and study and understand each of them, because they can either come as a group, as a long question, where you can discuss all 3, but they can ask you to discuss either one of these or any one of these. So make sure you understand which one is which and don't get confused. So for law 1, it's the principle of segregation. Okay, so what this is each trait, remember, whether it's in plants, the color of the flowers, how tall it is, where the flowers are situated, the color of the pods, the seeds, all of those things are traits. So each of those traits, one trait, is controlled by 2 factors, which we call alleles. So make sure you've gone through your terminology and understood what an allele is. So we've got 2 alleles that are situated on a homologous chromosome. And when gametes form during meiosis, remember meiosis is when we're halving the genetic material from either parent. So whether it's in humans or plants or animals, the mother and the father's genetic information is halved. Those alleles are split to create more of a variety when fertilization occurs. So the gametes contain one of those 2 factors from each parent. That's his first law of segregation. His second law is the law of dominance. Which thing is going to be more likely to express itself when we come to how things look, or the phenotype? So in the case of the plants, he cross-pollinated, and there should have been, equal opportunity, but it turned out 3-1, which means something was more dominant than something else. Okay, so let's read. The law of dominance says that certain alleles of a gene exist. Certain, not all, okay? Certain alleles exist in either a dominant or a recessive form. And if the pair of alleles are different, the phenotype, in other words, how it ends up looking, will only show the dominant trait. So that's why we get, when we've cross-pollinated the plants with a capital T, small t, and a capital T, small t, the capital T represents the dominant gene, which is for a tall plant, okay? And the small t is for a short plant. And we end up with a dominant trait here because 3 of the plants came out tall. So that's how the dominance works, and we'll get more into it as we go along. Okay, and the third law of inheritance is the principle of independent assortment. So due to random arrangement of chromosomes, because remember during meiosis, the chromosomes line up at the equator to go through the process very randomly. It's a completely random event, and so potluck really. And so any one of the 2 alleles of one characteristic can sort with any of another. And so that's why we get these variations. The alleles of different genes move independently of each other into the gametes, okay? So it's going to appear in different combinations, and that's the beauty of it. It makes genetic studies very interesting. Okay, so let's move on now to the genetic diagrams. And it's important to start with a pair of alleles in the mother and a pair in the father. Okay. So we know that we get half our information or genetic information from the mother or from the father. Now, we can have a situation where the pair coming from mom or dad, can be what we call identical or homozygous, or different, heterozygous alleles. So we do alleles with letters, capitals for the dominant and small for the recessive. In our process that we're going to follow, P is going to stand for the parent generation, which is the first generation. Well, not first, don't get confused, sorry. But it's the starting point, let's say, the parent generation. I'll explain something shortly. F stands for the offspring or the filial generation, and that's F for the family. F1 generation is the first filial generation. F2 is basically the grandkids. Okay, so if you think about it in normal human biology, you've got your grandparents who would, let's say, start off by being the parent generation. Don't get confused by the words. So grandparents are the parents. They create the first set of offspring, which is the F1 generation, which is your mom and dad. Okay, and then you become the F2 generation. But actually, the whole thing resets itself, because by the time you are at the level of having your own family, you become the parent generation, and those below you become the F1, and that's just how it goes. Okay, hope that's straightforward enough. Don't get confused. Okay, so we're now going to look at something called genetic crosses. So we want to know and learn the exact layout that they've got shown here, because in the exams, they give marks for all sorts of different things, and it's important that you follow the strategy and you do it structurally as they lay it out here. Okay, so this is the kind of diagram that they like. This is a genetic diagram to help you solve genetic crosses when we have fertilization occurring. Okay, so we start off with the P1 generation at the top, and we write phenotype. There's a space, times or crossed, and another space. Okay, and then genotype, a space crossed with, and another space. So this is how we're starting off. And once we have meiosis, we have the gamete, the result of the gamete, which we'll see. So fertilization, you can see the cross diagram there. We're going to end up with, in the F1, what is the genotype and what is the phenotype. This will make sense when we start doing examples. If you have a look at the explanation, a visible trait, like I've said, is the phenotype, which is the tallness or the shortness, or in humans, it's hair color, eye color, skin color, whether you're male or female. Things as crazy as if you have what we call dangle earlobes, or sometimes people have an earlobe that just goes straight across. It's not like down in a loop. The craziest example is whether or not you can roll your tongue. So as an example, I like being rude. So if you can do that with your tongue and do the curl, that is genetically inherited. And not everybody can do this, very strangely enough. And if you can't do it, you'll never be able to do it. You'll never be able to learn to do it. It's not something you can practice. It's genetically determined, and it's come from your parents. So either one of your parents were able to roll their tongue, but maybe not both. That's quite interesting. So if your parents are still around, go and check with them and see if mom and dad, who can roll the tongue and who can't, and it's quite interesting. Okay, so we also have another option to do where we can do it in this way. Similar, you have P1, the phenotype, genotype, meiosis, what the gametes offer, and then we have fertilization, where we have our square, okay, our Punnet square. And it's actually quite easy to use once you see, and we do a couple of examples. So remember that the alleles separate, okay, during meiosis to form the gametes. So if we just go back a tiny bit, sorry, if you look in the explanation box, the genetic makeup of an individual is its genotype. So whether it's 2 T's, capital T's, or it's a capital small or 2 small T's, that's its genetic or genotype, okay? The alleles separate during meiosis to form gametes, and each gamete has only one copy. So it's either going to have the capital T or the small t standing alone, okay? During fertilization, the person or the individual resulting is going to get one allele of the gene from each parent. And that's how we see the crosses starting to happen. Okay, so we'll move on now. This is also something you really need to get familiar with. We have to work out the ratio or the percentage chance of the various phenogenotypes occurring. For example, if there's 4 possible genophenos in total, only one having a particular phenotype, it'll be in the ratio of 1 to 4, or a 25% chance, okay? So then also a nice little summary here or recap. when you're doing exam questions, you need to read your exam question super carefully, okay? You need to take your time and really read what they're giving you carefully to find clues, and also so that you don't mix things up and make a mistake. Because if you make a mistake in one point, the rest following isn't correct and you don't get marks, okay? So take your time, be thorough and systematic when you're doing genetics. So first of all, which characteristic or phenotype is being examined? You have to understand what it is you're trying to look at, e.g. the shape of the seeds in this example here. Which allele of the pair is dominant? So in the case of the seeds with Mendel's pea plants, is it going to be round or is it going to be wrinkled? You know, for the color, is it yellow or green? One of them is dominant, one of them is not. It's recessive, okay? Also, we always ask ourselves, were the parents homozygous or heterozygous? Or are they both heterozygous? Because that will also make sure that we get the genotyping correct. And then we always ask what letters are being used, because if they didn't give us letters, we can select our own letters. But if they've asked you to use certain letters, please don't go and make up your own, because then it's going to not result in any marks. So be very careful which letters they've asked you. We always do a capital for the dominant allele and a small for the recessive allele, okay? We show how the alleles are separated during meiosis to form gametes, and then we show all the possible ways that fertilization can combine, which is where we get the crossovers between the sperm and the egg, okay? We distinguish the phenotype from the genotype. Great. So let's keep going. Hope you're still okay. I'm going to need to swig tea intermittently, so bear with me there. Okay, so when we're doing genetics, we always talk about a monohybrid cross, but we also get dihybrid crosses. To start off with the simplistic version, a mono always means one, mono, and di is two. Okay, so monohybrids refer to crossing of genetics where we only involve a single characteristic or trait, to keep things nice and simple. Obviously, dihybrid, it's two or more characteristics, so that's being handled separately. Okay, so when we handle monohybrid crosses, we are talking about dominance. Let's think about the thing with dominance now. Okay, we've got three types of dominance: complete, incomplete, and codominance. Again, very important that you understand the differences here and you don't get confused. And you've also got to read your questions in the exam very carefully to see if any of these words pop up by way of helping guide you when you're doing your problem solving and stuff. Okay, so we'll start off with complete dominance. What is it? How do we see it? So Mendel shows that monohybrid crosses, remember one trait, can have complete dominance. And incomplete dominance, it means the dominant allele masks or blocks the expression of the recessive allele if we have a heterozygous condition. Because obviously if we've got a homozygous, it's equal. It's capital, capital, or small, small, okay? So the following example is a genetic cross showing complete dominance. Let's work through it. Let's talk about it. For problem 1, he looked at the seeds and whether they were round or wrinkled. So this was just one trait. Remember, the plants had a number of different characteristics to look at, but he's isolating just one. This is monohybrid, okay? So we use a genetic cross to show the genotype and the phenotype of the F1. When two heterozygous plants with round seeds are crossed. So remember, the plants are showing that they're round, but if they're heterozygous, they're actually carrying the possibility of the other option. It's just not been shown. So either they're round or wrinkled, but when we see them, it doesn't necessarily tell us what's going on genetically. Okay, you sometimes have to figure it out by doing these experiments to see. An example of where they do this is, for example, in livestock farming with cattle and horses, all livestock that are bred for human purposes, really. But if you think about it, there are applications, and we'll get into that a bit later on why you want to know how this works and what use it is because of what you're trying to achieve in subsequent offspring. Okay. So in this instance, or in this example, the allele for round seeds is dominant over the allele for wrinkled seeds, which means it tends to happen more and come out more. They use the capital letter R for round to show it's dominant, and the small r for the wrinkled seeds because they are the recessive. We start off in our diagram, and this is how you would do it in your exam. P1, which is our parent generation. The phenotype, remember, it's how it looks, it's physical, what we see. We've got round seeds and round seeds, okay, because we took two heterozygous plants with round seeds. Now, here comes the important part, the genotype, what's going on at the genetic level in the genes. Capital R, small r, because we said they're heterozygous, which means they're carrying both possibilities. It's just that the capital R is dominant, and that's why we see the round seed. But it's still got the potential for a wrinkled seed in there. And the same with the other parents. So we've got our two parents with capital R, small r, crossed with capital R and small r. Now, remember when meiosis takes place, those R's are going to split because we're going to have one allele going off into each cell to create that possibility of we don't know which one we're going to get. So for the gametes, if you look there, we've got now meiosis happening, we've got gametes. So now we break it up into capital R and small r is separated, cross, capital R, small r. And now when you cross-link, because you've got to cross-link each thing with each thing. So if you look at the way the diagram, it looks like a whole bunch of squiggly lines, but it's quite logical. If you follow here, you've got the capital R, and when it crosses with the capital R from the other parent, in other words, capital R, it results in two capital R's. When we cross the small r with the other side of the large, or the capital R, we're going to get these results. Capital R, small r, and another capital R, small r. And the final result is going to be the two small r's combining, and we're going to get two small r's or recessives. So now in the F1 generation, very interesting thing has happened. We get our genotype, which is at our genetic level. So notice we've got one, what we would say, purebred homozygous. We've got another homozygous at the other end, and then we've got the two heterozyguses in the middle, which are similar to what we started off with. But now we've created the following situation where what comes out when that plant grows, we end up with round seeds because we've got two capital R's. We end up still with round seeds because we've got a capital R, small r, and remember the dominant one takes over and brings out that trait. So we end up with three round seeds, but right at the end, we've got two small r's. So the only expression they can have is wrinkled seeds, and we get this 3-to-1 ratio, which suddenly, the wrinkled plant pops out and everybody goes, wow, where did you come from? But it was always there. The potential for this was always there. It just wasn't always seen when the plants were growing. Okay. Now, the option that if you don't like that crisscross method with all the triangles going crazy and it makes you too dizzy, I personally prefer the Punnet Square method. So again, same thing. We've got the P1, our parent generation, the phenotype. They both looked like they had round seeds. Okay. The genotypes, we had capital R, small r, and our gametes separated as before. Now we carry it across into our little Punnet Square. We write the word gametes in the top corner, and then you represent one of the parents across the top. So that's the capital R in one block and the small r in the other block, and the other parent down the left, capital R, small r. And again, you cross both to get your result. So capital R with capital R gives us two capital R's, big R, small r, that block, the same capital R, small r. Remember, always put the capital letter first. So in this case, we get small r, big R when we cross. So we don't write it as small r, big R. We do it the other way around, okay? And the final result, two small r's. Same result, we get three round plants and we get a wrinkled plant. And the genetic or the genotypic ratio, this is now the percentage or the ratio, okay? We get one RR, capital RR. We get two mixed or heterozygous, which is capital R, small r, and we get one small r, small r. So it's a 1-to-2-to-1 ratio. And that's the genetic. And in the phenotypic ratio, the resultant thing that we see, the physical characteristics, it's going to be three round seeds and one wrinkled, which is a 3-to-1. Okay, so just note that very carefully. Don't get confused there, guys. So if we're talking about the genotypic ratio, it's definitely got to do with the genetic level. And you can see how that's a 1-to-2-to-1 ratio, because there are actually different combinations there. But the physical thing is only two choices, either round or wrinkled, 3-1. Okay, good. So a cross between two heterozygous parents would produce, because remember that's what we started off with, our two heterozygous parents, two equal genotypes with capital R, small r. When we cross those, it produces 25% homozygous dominant, which means the capital RR. 50% were heterozygous, which was the capital R, small r, and 25% were homozygous recessive, small r. Okay, so that's where the dominant and recessive comes in. Don't get confused with that. Okay, there's another activity that one can look at. I'm not going to cover that. There's the tongue rolling I was talking about. You can either do it or you can't. It's genetically determined. So it's potluck. It's the luck of the genetic draw. Okay. So now we've talked about complete dominance. We get something called incomplete dominance. And this is a little different, but it still works on the same principles. So how we do the crossing and the matching is still going to be the same, but the result is going to be slightly different. Okay. Tea time. Hold on. Okay, so incomplete dominance is going to be a cross between two phenotypically different parents, okay, which means how the two parents look. They're going to look different, and we're going to combine them, and we don't have one of the alleles being dominant or recessive. It's kind of like it's incomplete. It's kind of nobody wins, Everybody slugs it out, and I want my genes to dominate. No, I want my genes to dominate. And they can't agree, and in the end, the resulting offspring don't look like either parent. It's a different result, okay? But this is all to do with the phenotype, how it looks. So the offspring, the resultant offspring, is going to be different to both the parents, okay? It doesn't look like either one of the parents, and it's a new phenotype. The alleles have blended. They've kind of meshed together, and they've created a new phenotype, which is basically how it looks. And we'll see that. Let me scroll down and show you the example they use. It's the flower example. We started off with our parent plant being red and our other parent plant being white. And the result was pink flowers. So none of the flowers looked like mom or dad. Okay, there were no red flowers. There were no white flowers. They actually all came out pink. They were kind of diluted and mixed because neither one of the alleles for the color was going to dominate. No matter what the genetics was saying, neither one dominated. So we'll go back quickly and have a look at how that worked. So we had a homozygous red plant with a homozygous white plant, okay? And it produced pink flowers. So let's do the same thing with the Punnet square. As I said, I prefer the Punnet, and I think it's probably easier. So here's our genetic diagram. This is how it would go. We start off with our parent generation. What is our phenotype? How does the plant look? It's pure red for one parent, pure white for the other, and genetically represented by capital R, because those are pure homozygous parents, and capital W, again, homozygous, okay? During meiosis, we get the split of each allele. So we've got one R over here, one R, and another W and a W. When we put it into our Punnet square during fertilization, those have to come together and combine in the various forms. So we've got one parent across the top, which is the red one, R and R for its two gametes. And down the left side, we've got the parent who's white, W and W. Now, when you cross each block together, capital R, W, capital R, W, capital R, W, capital R, W. And so, because they are both dominant, you can see they're all capitals. We don't have a small for a recessive option here. They're all dominant. The resulting phenotype is pink. It kind of washed out the white and the red, and it didn't allow for that to express. So this is incomplete dominance, okay? And it's all capitals here. So the characteristics investigated were flower color, and the alleles are red and white, okay? In the flower example. And there we go, that's the result. So they now give us another example with mice and a couple of other things. Again, if you download this, you can go through these activities. I'm not going to cover those now. Okay, so let's move on to codominance, not to be confused with incomplete dominance, okay? It's a very distinct difference. So in codominance, both alleles, okay, of the gene are equally dominant, and both are expressed in the phenotype, as opposed to when they disappeared and got diluted. Okay, so this is an example of codominance. If we look at this, it's going to be the example of the cows. So you can see that the mom and the dad are purebred or pure colors, white and red cows, and the resulting kids, the little calves, are all pure red and white. They don't blend into a different color. They actually show dominance of the red and dominance of the white equally. Okay, so codominance. Make sure you understand the difference, and always refer to examples and pictures to help you understand this. It just makes it so much easier. Okay, so for the cattle example, we had that there are 3 color variations possible: red, white, or red and white, okay, in patches. So we get the red allele, which is capital R, the white allele, which is capital W, and we go ahead, we do our Punnet square. For our first parent generation, we've got a pure red-coated animal, which is capital RR, and a pure white-coated animal, WW. We do the meiosis step where the gametes separate, we put it into our Punnet square, and we cross-match everything, okay? We end up with our F1 genotype, which is the capital R, capital W. Notice again, no small letters here. But the phenotype, the resultant babies, how did they look? They were all red with white patches, okay, all. There were no babies that were pure. So once we combined these 2 parents, there were no babies that were pure white or pure red. They all came out with red and white patches, and that would be codominance, okay? Now, the characteristic being investigated was coat color, controlled 2 alleles, and the offspring produced the phenotypes as we've discussed, okay? So the offspring have a genotype. Remember, the genetics of the offspring are capital R, capital W, which is red and white patches. Now, it would be quite interesting to see what would happen if we took some of the offspring. Bear in mind, they now carry the RW genotype, okay? And if you go and cross them, go and do it for yourselves and see what result you get. Are we going to end up with red and white patched babies, or are we going to end up with possibly a pure red baby or a pure white baby? So go and do that for yourself and see what result you get. Right, okay. The next problem or genetic scenario that we sometimes get in the exam is sex determination, whether offspring will be male or female. This is obviously not in plants, but in animals and humans. So we have a problem here that we can work through. A couple has 3 sons, and the woman is pregnant again. So show by means of a genetic cross what the percentage chance is of the couple having a baby girl. We go through, we know that for humans, obviously our phenotype is a male and a female. We know that what that looks like. And the genotype for the males, they carry the sex chromosome, which will determine male or female in their offspring. XY, the female carries 2 X's. So during meiosis, again, those gametes separate. We get X separated from Y, and the female has 2 X's. So you'd think at this point, we've got 3 X's going on in this equation and 1 Y. It's going to be a little unfair. It's going to be a little, not balanced. But actually, genetics and biology is quite incredible, because have a look at how a process unfolds. During fertilization, again, we write the one parent across the top, the male, XY, and the female's gametes are down the side. And when you cross them, X to X, X to Y, X to X, X to Y. And the result, it speaks for itself. We get a 50% chance of a male or a female. But notice that the male does the contributing of that Y gene. It's not the female. So it gets a bit controversial, but yeah, when dad's blaming mom and saying, why can't you give me a son, or, whatever, it's dad that determines if there's a son or not, because mom only has X chromosomes. Dad is the one who has the Y. And so it's just the genetic combination of which one comes out. It's luck of the draw. Okay. So it shows the percentage of having a boy or a girl is 50%. And notice how they write the phenotypic ratio, which is the expression, the physical expression, 1 to 1. Okay. In humans, we have 46 chromosomes. So this is something to take note of because this is, again, potential exam question. 46 pairs of chromosomes. We get 23 from mom and 23 from dad, okay? Of those 46 chromosomes, 44 of them are going to control our appearance, our structure, and our functioning of our body and all of those things. Those are called the autosomes. And the remaining pair determines the sex of the individual, called gonosomes, okay? Which is quite amazing. Just that one little set of chromosomes will decide if it's a boy or a girl, okay? Each species, in other words, humans, have a specific unique number, shape, and size of chromosomes. That's called our karyotype. In animals, it's different. Just as an example here, a karyotype 1, we know that the 23rd pair of chromosomes is the sex chromosome. So in this example, if you look at pair number 23, would you say that that's a male or a female? And in example 2, it's different, male or female. So you can see it's female in the one and male in the other. Okay. So, right. I'm going to stop the video at this point, and I'm going to pick up the next video from this point. We'll carry on here where we're going to stop, because I'm just going to take a quick break in the next one.